Chapter 3 of our behavioral neuroscience textbook was all about homeostasis. And so segment 3 included pages 417 to 425 and first started talking about how we can store energy for future needs. So complex carbohydrates, which are found in our diet, are our most immediate source of energy for our bodies, and they are rapidly broken down into simple sugars called glucose that cells can use. So glucose can be converted into glycogen to be stored as a reserve fuel in several locations, especially the liver and skeletal muscles for short-term storage. And the process of the conversion of glucose into glycogen is called glycogenesis, and it's promoted by the hormone insulin. So glucagon mediates the conversion of glycogen back into glucose, and this process is called glycogenolysis, which is triggered when glucose levels drop too low. And that's for short-term storage, but for long-term storage, lipids, which are large molecules of fatty acids and glycerol that are insoluble in water, they are dropped in fat cells that make up the adipose tissue, and these serve as our long-term storage of energy. And fat can be converted into glucose through gluconeogenesis, or a secondary fuel source called ketones. The next section talks about insulin and its role and how crucial it is for the regulation of body metabolism. So insulin enables the body to use glucose by breaking it down into glycogen, and most cells regulate the import of glucose molecules via glucose transporters, which are molecules that bring glucose molecules into the cell for use, and they must interact with insulin in order to function. So there are three primary sequential mechanisms that stimulate insulin release in the body. The first phase, which is called the cephalic phase, is mediated by the brain and occurs when sensory stimuli from food evoke a conditioned release of insulin in anticipation of glucose arrival in the blood. And then the second phase, the digestive phase, occurs when stomach and the intestines release gut hormones when food enters them, and some of these gut hormones stimulate the pancreas to release insulin. And the third phase, the absorptive phase, occurs when special cells in the liver called glucodetectors detect the glucose entering the bloodstream and signal the pancreas to release insulin. And so information from the glucodetectors travels via the vagus nerve to the nucleus of the solitary tract, or the NST, in the brainstem, and is relayed to the hypothalamus. And so a lack of insulin can cause the disease diabetes mellitus, which is the excessive glucose in the urine. And there's two major types of diabetes. Type 1 involves the pancreas stopping production of insulin, and type 2, which is when there's reduced sensitivity to insulin. And despite their importance, neither insulin nor glucose is the sole signal for hunger or satiety. Hunger meaning the internal state of an animal seeking food. Lower insulin levels can cause hunger, but again, it's not the sole source. And high insulin levels direct much of the glucose out of circulation and into storage, which results in what we call hypoglycemia. The next section talks about the hypothalamus and how Several systems control hunger. So the dual center hypothesis involves two major parts of the hypothalamus. The first one being the ventromedial hypothalamus, and lesions of this result in animals eating to excess, a behavior we call hyperphagia, and thus the animals became obese, which implicates the VMH in the satiety center. And the second part of the hypothalamus, the lateral hypothalamus, is implicated as a hunger center because lesions cause aphasia, which is a cessation or stopping of eating and rapid weight loss. So multiple peripheral signals are integrated by a hypothalamic appetite network. So the arcuate nucleus of the hypothalamus is implicated in appetite control. Other information about energy balance, especially the short-term and long-term reserves, come in form of hormonal secretions from elsewhere in the body. So there are three major peptide hormones we talk about here, and the first one is leptin, and it's released by fat cells. It's found in the chloride, plexus, the cortex, and several other hypothalamic nuclei. Defects in leptin production or leptin sensitivity can cause a false underreporting of body fat and can lead to overeating, especially high-fat or sugary foods. The second peptide hormone is ghrelin, and this comes from the gut, and it's an appetite stimulant. So levels rise during fasting and drop after eating. And the final peptide hormone is PYY336, which is secreted by the intestines and probably acts on the hypothalamic appetite control mechanisms to suppress appetite, as lower levels are associated with obesity. The arcuate appetite system has two sets of major arcuate neurons with opposing effects, and they are named after the transmitters they reveal. So the first set of neurons are the POMC neurons, as they produce pro-opiomelanocortin, or POMC, and cocaine, and they act as satiety neurons and inhibit appetite and increase metabolism. The NPY neurons produce the neuropeptide Y, or NPY, which may carry signals for feeding and hunger. So projections from these two types of neurons also leave the arcuate and make contact with neurons in other hypothalamic sites. The arcuate modulates food intake in this way. And another important thing to note is that leptin suppresses hunger, but by different ways in the two systems. In the POMC, high levels of leptin activate the appetite-suppressing POMC neurons, but in the NPY system, high levels inhibit appetite, increasing NPY neurons. So the second order hypothalamic neurons integrate appetite signals, and primary targets of the projections from the arcuate are two hypothalamic sites. The first one are orexigenic neurons, which are neurons of the hypothalamic appetite system in the lateral hypothalamus that promote feeding behavior. And then the second one is anorexigenic neurons, which are neurons of hypothalamic appetite system in the paraventricular nucleus, which also releases oxytocin and vasopressin. And this inhibits feeding behavior. And one set of projections from the appetite-suppressing POMC neurons of the arcuate terminate in the PVN release alpha melanocyte stimulating hormone, or alpha MSH. And this is a peptide hormone that belongs to a small family of substances called melanocortin, which are derived from the POMC, and they act on the melanocortin type 4 receptors, which are a specific subtype of melanocortin receptors, and they activate PVN's appetite suppressant activity, resulting in a net decrease in feeding. The NPY neurons inhibit anorexigenic PVN neurons and thus promote increased appetite, and the AGRP release by these neurons into the LH compete with the alpha MSH hormones for their receptors, which counters the appetite suppressing influence from the POMC neurons. And a final substance that plays a role in this system are called orexins, which are also known as hypocretins, and they are neuropeptides that are produced in the hypothalamus, and they are involved in the switching between sleep states and the control of appetite. So there are several other systems that play a role in hunger and satiety, and appetite signals from the hypothalamus converge on the nucleus of the solitary tract of the brainstem, and information about nutrient levels are conveyed from the body to the NST via the vagus nerve, which we briefly touched upon earlier. The peptide cholecystokinin, I think that's how you say it, is released by the gut after feeding, which acts directly on receptors of the VN to inhibit appetite and thus serves as a signaling molecule in the brain. And the pleasurable aspects of eating, meaning like the flavor and just enjoying a good meal, the amygdala and the mesolimbic dopamine mediated reward system coordinated with the LH has been implicated. Another point to take is the endocannabinoids, which are the endogenous ligand of cannabinoid receptors. These ligands can potentially stimulate hunger by affecting the mesolimbic dopamine reward system as well. And those that stimulate the POMC neurons selectively activate the release of beta endorphins, which play a role in reward mechanisms, and not the release of alpha MSH, which plays a role in satiety.